So what does that end stage look like? You got to begin wherever you are in your process. If you're a small biotech that's taking technology out of academics and you're ready to start clinical trials and get into that process, you've got to know what that end looks like, and you've got to start planning early for that. So again, it's about what are the expectations from investors, from regulatory for that end stage. So I like to talk about mapping it out, and those of you that have worked with me know that I love mapping things out, probably to the extreme, but if you don't know where you're going, it's hard to develop the strategic pathway to get there. So I think it's important to early on develop a pathway and look at strategies that are going to help you get there, understand what the BLA expectations are, and understand what the market looks like for commercialization. That's especially acute focus right now for cell and gene therapy. A lot of us are at that critical stage, but we've got to have a product that's very commercializable and appealing to investors to take us to that next level. So you have to, one of the things that I found in working with companies and talking with people is not planning effectively ahead. It needs to be a process that you can move forward and you understand where you are, where you're going to go, and have plans on how to get there early on so that you can be effective in your time management, your resources managements to get to those stage. Some of the critical components of that is early integration of quality systems. That's really critical. Data integrity and documentation that'll take you forward in the manner that it has to be submittable to the regulatory agencies. And obviously, developing scaled and cost-effective manufacturing. So kind of one of the critical points is you start with knowledge gathering. And I'm making a broad assumption that you're starting really early. So you need to know what the BLA requirements are. This for us in this field right now, and mostly in this audience, it's going to be about CMC. One of the things that I find very interesting, AABB just had a conference in Houston, and an FDA speaker was there, and he was talking about the field of cell and gene therapy and all of the submissions to BLAs. 65% of the issues were all CMC. The rest were clinical. And a lot of them got pushed back for significant periods of times, year, 2 years, because they didn't have it ready for a BLA CMC section submission. So want to bring some of the things that you need to be thinking about in your preclinical phase 1, phase 2 studies, that you need to think about what it needs to look like for that BLA. And it starts with building in quality systems out of the gate. There's a lot of resistance with CEOs and CFOs to put a lot of money and time into that effort, but it is critical. Your product is only going to be as good as the quality that you build into it, and building that in early as per FDA or any regulatory expectations is hugely important. And work with your senior management. What are their perspective times for market? What are their, when do they want to submit? When do they think they can commercialize? And then work backwards, build it into your timeline, and force them to be realistic about it. Investors and CEOs can be really pushy sometimes. Boards can be really pushy and say, well, you can do that in a year. No, you can't. So, but again, it's about build your knowledge base, know what it's really going to take to get there, and be realistic and work across your disciplines to get it occurring. And last but not least of that, you've got to also gather and buy corporate and board and investor investments into your buy-in of what your path forward is. You can't ignore them. Coming a little bit more from a science and loving the science behind what we do, it's easy to get too excited and delve into that. But when it's all said and done, it's about getting that product to market so that we can get a product that can potentially help patients sooner than later and get the resources to get that done. So we have to get their buy-in also. We won't go into this in detail, it'll drive you bonkers, but I map everything out. Again, it's like, and it's a living document. It's like, what do I think I need? What are the components that are relevant? And what are the stages that you need? And who do I partition it out to? I've got this folded up quite a bit, but it's quite a big map. And then I've got subsection maps of this that we work from. But today I want to bring to focus about the CMC aspects of a BLA. Obviously that CMC submission know, look, see, learn what's expected for a BLA submission. A huge contributor to that is obviously your process development and manufacturing and how you go through that process in a fluid manner. And you cannot forget the quality aspects. There's a lot of aspects of that that are relatively important. I could talk all day long, probably all week on all of the aspects, but I'm going to focus today a little bit on the concept of assay and process development and qualification and validation, and just a teeny tiny bit on how important your documentation is. So there's a term out there that gets used a lot. It says phase-appropriate development and validation. The tendency is, well, okay, we can do that at stage 2. Okay, we can do that at stage 3. Well, by the time you're at those places, it's a little bit too late. So it doesn't mean you can push it off, and there's a tendency to push those critical components off. But it goes back to understanding, what can you build in as you're starting your phase 1 trial, phase 2 trials? What do you want to achieve? And what do you want your pivotal phase 3 study to look like? Because honestly, that's what you're really taking to your BLA. And a little quick lesson learned listening to some of the quality people at Kite. I don't know if any of them are here or not, but they've had some customer feedback. What about this? What about that? Once you put it in a BLA, it takes a monumental effort to change anything. And I've heard them repeatedly tell me, I wish we had done this earlier. I wish we'd put more time in what we really needed it to look like at commercial lines. Of course, they had the pressure, get it to market, get it to market. Novartis had beat them. They got to get right there to them, too. So it's a hard balance, but that's why the sooner you can start building those elements in, and it's easier than it is perceived to be. So I think that's usually important. But it's inclusive of a lot of things. It's inclusive of your assay development and your process development, how you guarantee that quality is built in from the very beginning all the way to your final pivotal phase 3 study. So I've lined out some things. Ooh, that's small. I can't read it. Again, I don't want to go through all they see it bigger, okay? I don't want to go through all the details, so you can take the time to look at this a little bit more. But there's critical things to think about before and during your phase 1 study. Something that gets pushed off to clinical all too often and ignored in manufacturing and MSAT labs is that target product profile. You need to be integrated into what clinical thinks it needs to be, what your hard R&D scientists think your mechanism of action is. You've got to understand it, and you've got to contribute to that TPP. That's another very much so living document. What you know today is definitely not what you're going to do by the time you get to the end of a phase 3 study. You've got to start building and understanding your quality attributes much sooner than later. And that's more, again, about having a consistent, reliable, reproducible product and being able to take that forward, so you can't bring those in any sooner. I mean, the sooner the better. Obviously, you've got to bring the safety testing in. That's pretty standard for us in the industry. But there's a lot of process controls that you need to go through. And then before and during phase 2 is where you really have got to do a lot of that optimization of your process. Know what you really want your pivotal study to look like, i.e. what you want that to look like. So I'm going to quickly go through some of the important things that I think are involved in that. So you've got incorporation of quality systems, integration of automation, and good documentation practices. So there's a lot of core concepts with manufacturing and bringing in quality systems. You got to make sure that you've got a sustainable process, that it's reliable, reproducible, and it's cost effective. So I'm going to skip through this. It goes through, another process map. But the bigger things about that is understanding the stage of assay development. Kind of during preclinical phase 1, phase 2 studies, you need to understand the design, its fit for purpose, and the qualification. And then during your phase 2, phase 3 studies, you got to really build out that characterization, really understand your product inside out, its potential functionality, its potency potential. And then during that phase 3 study, you have to validate all of your assays, all of your process. It has to be a complete package to submit to your BLA. For me, and my entire history in this, I've had more success with starting out from the get-go, developing internal process controls. We run a tissue bank of PBMCs. We run a tissue bank of our seed material for manufacturing. We run a tissue bank of our product with consistent donors that anytime I make a change, I know historically what those are going to do, and I can really do very effective comparability studies. There's no commercially available controls for what we do, and I can't stress the importance of building that early, and it helps streamline and makes for very efficient comparability studies as you make process and/or equipment changes. The next thing about that is, fine, you've got these controls, what are you going to do with it? You got to look at your data. You've got to see what it's saying. I like to take it from the aspect of an assay. It's a little bit more straightforward. You've developed this internal process control. You expect it to do X, Y, and Z. But how do you use that to qualify your assay to say you've got releasable data? There's the classical Levy-Jennings charts, but there's a concept out there that I learned from the clinical world called Westgard rules. And it's a really good tool that you can easily build into charts. And it's going to say, I've got an outlier. Is that a releasable product, or is it true OOS, things like that? Incorporate that early. That's going to help build out your qualification/validation package that you submit. My favorite topic is automation. I won't, again, go through this, but we automated early. We had a lot of intention, largely driven from an investor standpoint. How can you get me there faster? How can you reduce my cost of goods? So again, there's points to consider in that. And I'll be glad to share these slides with anybody. But we started out quite early. Our current phase 2 ready for our HER2 study. We've incorporated the LOVO to clean up our Luke. We use the Clinimax right now to select for our target CD4, CD8 cells, and then we use the Cocoon for the entire manufacturing process. We did our phase 1s with the Cocoon. For the phase 2, we brought in the LOVO and the Q for our fill and finish. Is that where I need to be for commercialization? Not quite. So we're looking at doing process development using those known donors as process controls to look at things to make that a much more fluid assay and what I wanted to look at for BLA. So a few key takeaways there. I want to emphasize pretty much I lightly talked on all of it, map it out, but communicate with all the players at the table. You've got to understand their perspective. You've got to understand how it fits in what you're doing, and document. Lesson learned, I went to a end of phase 2 study, pre-phase 3. It's amazing what documentation the FDA wants. You better have built that along the way. Going backwards 4 or 5 years is a nightmare, so build it as you go. Have a great documentation package. And my buzzwords is fit for purpose, data integrity. Your product is only as good as your data. It's quality data. If it's not good quality data that you can stand behind, you don't have a product. Use those internal process controls that can really help you build forward. And get quality integrated into your system from day one. Stress that with your senior management. And again, that comprehensive documentation and map it out. Know where you're going to go and what you got to do to get there.
Thank you very much, Donna. Our second speaker is David Hermanson, Principal Scientist at Cell&Gene Therapy for Bio-Techne. And his presentation will be closed system, open possibilities, consideration for future-proofing cell therapy manufacturing.
All right, thank you, and welcome to the session this afternoon. So we just heard a great talk about thinking about the quality and thinking about that right away from the beginning. I'm going to focus more on the actual process steps and what you can think about from the get-go when you're actually building your manufacturing process. So in order to put this talk together, I started to lean back on some of my process development days. So I used to be a process development scientist, and I've built a couple of CAR-T manufacturing processes, and I've never gotten it right. There's always been things that I wish, and I look back on, that I would have changed. But I was able to kind of pull out a few different keys that I think you should think about right away off the get-go that can help you to get it right, or at least as close to right as possible. So the first of those is understand your options. So I started in this field a little over a decade ago, and there were limited options, right? We didn't have all of the stuff that is out there in that exhibit hall. today. We didn't have that 10 years ago. And so if you wanted to, you could sort of brute force through everything and you could test everything that was on the market, figure out what worked the best and move forward. That's not practical today, right? And we'll get into that a little bit more. But you really do need to understand what's out there, what's going to work for you, and identify what's going to be the best fit. The second key is choosing your partners wisely. I think almost everyone who's been in cell therapy for any period of time probably has a horror story as to, when things went wrong, there's road bumps along things, and you got to have partners that are going to work with you, that are going to adjust with you, grow with you, right? There's been a lot of emphasis already at this conference about being able to address the actual patient population and commercialization. And so how are those partners that you're choosing out at R&D stages going to grow with you and be able to provide reagents, bioreactors, manufacturing platforms, whatever it is that you've partnered with them on, how are they going to do that? And then the third one is apply Occam's razor. And so Occam's razor is the easiest explanation, is probably the best. And so really just keep it simple, right? When you're developing your manufacturing process, the more complicated you get, the more fail points you have, and the harder it can be to realize that commercial success. So by the time you get to commercial, your process needs to be relatively simple. You got to have the data for sure, but it's got to be relatively simple. So again, taking a look back to 10 years ago, kind of if I break down a typical CAR T manufacturing process or any cell therapy process, you've got your starting cell population, you might have selection in there. Then you've kind of got your reagent pool, which is going to be media, cytokines, some sort of activation reagent. A lot of them are going to be gene edited, so you got to choose a gene editing strategy. And then finally, you've got your expansion vessels or your manufacturing platform and the fill finish. And so 10 years ago, again, when I first started, there were relatively limited options. And I think everything on that slide is something that I tested in process development when I was doing it and building a CAR T manufacturing process. Compare that to now, and I'm sure this list is not completely exhaustive, but if you compare it to now, there's more things than you could ever possibly try. If you were just starting out and you were going to build a process and you went and you tried to try everything that was on the market, you'd go broke before you got started. So how do you do that, and how do you really evaluate? And so with so many options to choose from, how do you make those decisions? And they're very critical decisions, and they're important to get right from the get-go. So how do you do that? So to try to simplify it somewhat, it's a complicated thing to do, but to try to simplify it somewhat, I've kind of broken it out here. And so we have manufacturing platform solutions. Things that you want to be evaluating from the get-go are the scalability. And everybody thinks scalability is up and out, but remember that you also need to be able to scale down, right? You've got to be able to do your R&D reasonable. You can't necessarily take a full leukopak for every run, even in your process development. So how do you parallel path things? How do you do test multiple conditions all at once? So you've got to be able to scale down as much as you do up and out. I think flexibility is key. So this is still an evolving field, and so remaining flexible is going to be key. Again, you're going to encounter roadblocks. You're going to find things that aren't working for you, and you need to be able to pivot and be flexible. So if you lock yourself in to a particular manufacturing platform, you've got to be able to adjust. And so sometimes that removes some of your options. And the more options you have, especially as you get closer to entering those clinical trials, you have to kind of apply the learnings, the better off you'll be. There's other things like all-in-one versus the modular approach, how complex the systems are. And then the other one that I'd like to stress is kind of a maximum utilization of equipment, right? So if I'm going to buy an expensive piece, I want that expensive piece of equipment doing its specialized function as much as possible. And so if you have a selection instrument, you want that thing selecting. You don't want to necessarily be incubating cells on it. You want it selecting as much as possible, as frequently as possible, because that is what you paid for, is for it to perform that part of the process. When it comes to reagents, you got to look at lot-to-lot consistency, GMP availability. This is, not as much of an issue nowadays, but again, going back 10 years, there are all kinds of reagents that were still in the RUO stage that were coming GMP in the next year. Developers would buy into that, they would start using them, and then GMP didn't come along for 3 or 4 years. And that kind of puts you behind the 8 ball, presents its challenges. And so nowadays you should really be looking for reagent providers that already have GMP capabilities. And then closed system compatibility, right? Everyone wants to close down their process, build in automation. And I think one of the key gaps that still exists is there's lots of manufacturing platforms that are fully closed, but not all reagents get provided in a fully closed format. So a lot of your selection beads or activation reagent or cytokines are still in a vial of some sort that you have to go in, use needles, and extract. When it comes to gene editing, you've got cost and complexity. Viral certainly still dominates the market. However, there are true competitors now with either transposons or nucleases, and going site-specific. So non-viral is a real thing that you can choose to do. You don't have to be locked into a lentiviral vector. And then this is another one, performance, right? You've got to decide from the get-go what biology is important to you. So you can't get lost in getting too fine-tuned with your manufacturing process that you lose sight of the biology and the efficacy. Because if you don't have efficacy, then you can have the best manufacturing process in the world, and it doesn't mean anything. Tip number 2, choosing the right partners. And I think this one's probably really crucial overall. And it's much like everything else in life. It's built on trust. It's built on relationships. And so I think that is really where you need to focus. That's a difficult thing to do. There's a lot of vendors out there, right? And every vendor out there will tell you that they have the proper quality systems in place and that they're the partner that you should be choosing. And so how do you build those relationships? How do you identify that? There's a couple of things I think you can do ahead of time. Look into their quality systems, browse their websites, make sure that they have GMP reagents available for ones that you're considering buying from them, look at the formats they offer them in, things like that, and then engage and have conversations. And then finally, keeping it simple, right? So avoid introducing any potential fail points, reduce the number of touch points you have and the interventions that you have to do. I'm a big fan of automation, but only where it makes sense. I think it makes sense to automate your selection, and a lot of other points, but it doesn't make sense to automate every single thing, especially early on. You want to build and future protect so that you can move more towards automation, but you don't need automation from day one. Although I think, again, there are certain points where automation should be applied early. You want to plan for success. A lot of the systems are still being released. In the last year, there's been 2 or 3 manufacturing platforms that have come out. And so, and I expect continued development in the area. And so maybe that perfect solution isn't out there yet. So how do you remain flexible so that if a perfect solution does come up, you can move to it relatively easily? And then again, I'm going to press the point of identifying what makes your therapy unique and then lean into that. So again, this goes back to that biology. Not everything. There used to be a saying in cell therapy that the product is the process and the process is the product. I don't really hear that as often anymore in my conversations. I think it's still somewhat true, but the processes are really not all that different. And it's really leaning into what is unique about your therapy and then finding and picking the reagents that support that unique biology. Because again, ultimately, I think you're going to live and die on the efficacy that you have from the therapy. And then the last one I'll point out here is collect actionable data. So a lot of things, I think we're still in sort of the age of data set building with some of the tracking as donors grow, what fails, what works. And so I look forward to the day when that data that we're collecting now becomes actionable, when you actually can then, modulate the culture in some way, add something, take something away, whatever, to turn something that would have been a failed process into a successful process. And I don't think we're there yet, and that's why I say collect actionable data as much as possible. So at Bio-Techne, just a couple of minutes on how we translate these learnings into practice. So we're really focused as primarily a reagent provider, creating fit-for-purpose solutions. One of the things that is relatively new for us is our Pro Packs. These are fully closed cytokine offerings in a liquid that are directly weldable into to like a media bag and can be delivered. And so they're a really great value add there on the cytokine front. We have GMP T-cell media. We have a transposon system called TC Buster, which does enable non-viral gene editing. And then we also have some characterization tools in our automated ELISA platform known as Ella, and then Simple Western for either lentiviral characterization or even CAR-T signaling pathway for mechanism of action studies. We've also partnered with Scale Ready. Again, I've been in this for a little over a decade. I've tried out a lot of different manufacturing platforms. I'm still a huge fan of the G-Rex. I think it is probably the best at that scale down aspect that I highlighted earlier. You can get it in the 6-well plate and you can scale it up and it scales out. So I think it's a great platform for incubating the cells and growing them. And then kind of leading with quality. So we've invested heavily into a manufacturing facility in St. Paul, Minnesota, dedicated to our cytokine manufacturing. And we've really leveraged our long history in producing proteins to ensure exceptional lot-to-lot consistency. So just like 30 seconds on a shameless plug. If you want to learn more about Pro Packs, I'll be talking tomorrow at the Innovation Center at 10:30 in the morning, just to kind of go over more about the Pro Packs and that weldable, fully closed cytokine delivery system. So just to kind of end here, this whole session is about beginning with the end in mind. And so what is the future of cell therapy manufacturing? So I think I've heard it already multiple times today, but I think we all acknowledge that cell therapies do offer the potential for real cures, but we haven't really fully realized that promise. A relatively small number, I've heard as low as 30,000, as high as 40,000. So, we'll just say roughly 40,000 patients have been treated with CAR-T worldwide. The number that could have been treated is far, far more. Yescarta kind of leads the way at about 4,000 patients a year. But as these therapies move up into earlier lines of therapy, that patient population is only going to grow, and that pool is only going to grow. And so if you don't have a manufacturing process that can, scale to potentially tens of thousands of doses a year, we're going to continue to struggle as a field, right? And so how do we get there? I also think as we start to see success in new indications, I still have a lot of promise that CAR-T will have an impact in solid tumors. And if that happens, a lot of the patient populations are much larger than they are in the hematological malignancy space. And so that's going to drive the need for more scale out and more high throughput production. And so to kind of wrap up my thoughts, the most successful processes are going to be the ones built to grow and expand to address the large patient numbers. And so what can you do? Maximize your equipment utilization, streamline your reagent handling, minimize your touch points, and remain flexible as you build that out. So I just want to thank you. I'm happy to engage further to talk about any of this stuff, but thank you for attention, and yeah.
Thank you, David. I'd like to welcome to the podium Kasia Averall, Vice President of Quality at Quell Therapeutics, and she's going to talk about the manufacturing feedback loops for autologous Treg cell therapy. Thanks.
Right, thank you very much. I'm Kasia, I'm from Quell, and I'm going to spend 15 minutes describing our journey in Treg cell manufacture from first in human to a process that we now believe is tech transferable to a CDMO. And really the focus of the presentation is knowledge management, what I've termed here manufacturing feedback loops. And I'd like to start, please, with a huge thank you to Austin at the back, who managed to reformat my slides after they went wonky crossing the Atlantic in about 5 minutes. So thank you so much for your efforts on that. And how do I move? Oh, yep. Okay, there we go. So a little bit about Quell to start. We were founded in March 2019, so about 5 years old, based on expertise from King's College University, University College London, and the Hanover Medical School. And our focus are T regulatory cells. These are a subset of T cells, and they downregulate the immune system. They quell it, if you will. So our therapies are gene-modified Tregs, which we are harnessing the suppressive capacity. Our lead clinical program has been spoken about a little bit already at different presentations, but it is there. It's Quell 001, and that is for solid organ transplant, and that is the program that I'm going to focus my talk on today. However, we have got an exciting pipeline coming up behind us. We've got a partnership with AstraZeneca, and we published a milestone on candidate selection right before Christmas. The partnership with AZ is in type 1 diabetes or IBD, so you can see that from a scalability point of view, we're really looking at kind of increased patient numbers in the future. So that's something we've had to bear in mind. So this is a high-level schematic of our manufacturing process. This is about as detailed as our IP department would let me go. But for those of you who heard Natalie Belmont speak just before this talk, she gave a really nice outline of some of the cell selection procedures. So we start with a human of indeterminate age and gender, because I can't tell you anything about the patients in our trial. And out of the patient comes leukapheresis, and this is our starting material. So we go through a cell selection step, which gives us the T regulatory cells that we want. Then we introduce the Quell's transgene, and we're using a lentiviral vector. Then a cell expansion of the now CAR Tregs. This gives us our final drug product. It's an autologous product, so it goes back into the same patient. And we have run this process a lot at Quell. We've done more than 100 sort of full-scale manufacturing runs between our GMP labs, our MSAT labs, and our PD laboratories. And these are both healthy donor and patient material. So we've had a lot of experience in improving this manufacturing process. So if I just go into that slightly, this is showing you some of our process development. And the process goes from process 2 to 2.5 to 2.7. So the first thing you want to get right is decide how you're going to number your process improvements as you go through. But you can see between process 2 and 2.5, in terms of our development, we were really looking at increasing our dose by increasing the transduction efficiency. Between 2.5 and 2.7, that's the right-hand side there, you can see that we focus on the expansion capacity, but we're retaining that phenotype. So what we have now is and I'm going to touch wood, a nice stable process that's yielding high doses. So for Tregs, we're making more than 1 billion cells. We can get a dose of more than 1 billion. That's a lot for Tregs. So it's a good phenotype, it's a stable manufacturing process, and it's a really high dose. And we got there really through process development. So when you think about your product, the dose and the phenotype are impacted by 3 kind of big buckets of variation. There's the patient, and we know about the patient, right? This is kind of the big thing in autologous cell therapy manufacturing. It's a very variable starting material. So the patient's age, their gender, their treatment, their immunosuppression regime in our case, their lifestyle, their apheresis, all these things can impact the drug product dose that you have and the drug product phenotype that you have. And it may be that you want to do some work with your clinical team, your monitoring teams, to look at what are the different factors that there are at clinical sites that produce a really nice starting material that give me the absolute best mechanism to get dose. And then maybe you can incorporate those into your protocol. But if you do that correlation, you're going to completely ignore the fact that actually maybe some of those doses and phenotypes of the batches that you're looking at have been produced under different processes. And so you've got your process variation as well. You've got your variation between the different process iterations that you have, and you have the variation inherent in that process. And the process variation really tells you what patient population you can access. So when we have done our process improvement, going from 2 to 2.5 to 2.7, we've broadened the patient pool that we're able to access, right? We've got a larger range of starting materials that we can take in and use to make a dose. So you've got a drug product, you've got a dose in your phenotype, but it's being acted on by the variation within the patient itself, and then the variation inherent in your manufacturing process. And then you've got my favorite bucket of all, because I came out of GMP, the operations variation. And this is basically your ability to execute a BMR time and time again, doing exactly the same thing. And I'm going to assume, because you're here in the title of the talk, you're fairly early stage. So these processes are probably fairly manual and fairly operator dependent. And so there needs to be a mechanism within your company, and I'd encourage you to think about it early. How on earth do you differentiate a batch that hasn't performed the way that you thought it would from something like operator A, for example, was doing something slightly differently, to actually maybe one of my cell selection materials wasn't performing the way that I thought it would, to, do you know what, that process, that patient wasn't going to happen in the first place anyway. So the answer really is it's a knowledge management system, and it's about going company-wide. So it's making sure that when you are looking at the data that you get as a company, and Donna really kind of highlighted this nicely in her talk, you're not just focusing on a little pocket of GMP and operations and doing it right, and you're not focusing in a completely different building and mindset with your process development team, and your clinical team don't talk to any of you. It's bringing it all together to make sure that you're getting a really good idea of what is impacting that drug product dose and phenotype, because otherwise you won't be able to do the good process development. So if I go into that slightly, one of the first things you can do and should do early on is to really reduce the operations variation. And I've shown that in a very sophisticated way. I made the operations ball get smaller. So this is the GMP side of it, right? This is the absolute bog standard, just do GMP manufacturing. And for those of you who've heard me speak before, I do harp on about it, but GMP is a discipline in and of itself. It's lovely having these amazing PhD scientists who can do flow cytometry really well working in your manufacturing clean room. But GMP, just being able to do something right, being able to execute a batch record, that is a skill in and of itself. So you can do things to make it right. And every time you have a failure to follow the process, a deviation, if you will, each time you have a mispick, or you expose your cells to a cleaning reagent, or you're waiting for a new QC analyst to give you the data so that your process takes a little bit longer, or anything like that, you're introducing variation into your operations. And it might sound a bit counterintuitive to start talking about standardization and GMP so well in your process, but let's consider you've got a clinical site and they are doing something that adversely affects your starting material that gives you a poorer drug product at the end of it. But for whatever reason, every single batch that has come out of that clinical site has been subject to some kind of manufacturing deviation. You've masked it, right? You're going to go in, you're going to do root cause analysis, you're going to say this batch fell because of ABC, completely masking the fact that you've got a clinical thing happening here. So you need to really reduce that operations variation in order to give yourself some more information. And that then makes it clear that your operations, it's not just making a drug product, it's really feeding knowledge to the rest of the company. And this is kind of what I'd encourage you to do. When you are setting up your knowledge management system or your business management system or your quality management system, whatever you want to call it, don't think about it just as producing a compliant product. Compliance, and I'm quality here, it's not the be-all and everything, right? You need to start also generating knowledge, because this is kind of where your company's success lies. So each time you execute a batch perfectly, you follow the BMR. The BMR is lovely and easy to follow and well thought out, and your operators are trained, and the process happens really nicely, and you still don't get a dose. That's information you can start to feed back to the rest of the company. And this is where kind of like these little feedback loops come. So I've got a very complicated schematic here. Manufacturing process A is running, and out of it is funneling the kind of information that your development department need to really start moving the process on to the next iteration. And then your development department have got a whole pile of really interesting information that can impact GMP, right? So it might be that you're changing your gating strategy, you're optimizing it. You need a way to translate that into GMP. You don't want your GMP operators doing too much on their own because you're reducing the variation there. So how do you kind of keep that kind of connection really tight? Equally, you have equipment. Now, the operating parameter of the equipment might be this wide for the manufacturer, totally fine, it's calibrated, it's qualified, you know, it's serviced, all the rest, blah, blah, blah. But actually, for your process, you need the operating parameter to be much narrower. This is kind of where your PD department really come in. They need to link that back to manufacturing. And if you carry on with this process, iteratively going back, pulling out all the information, you go from manufacturing process A all the way through to manufacturing process N, and you end up with where we are now. This press release to say that we've now expanded our manufacturing capabilities through a CDMO. So we did all this stuff in-house, and that was great, but we've learned a lot. We now reckon we've got a platform that is robust, scalable, and tech transferable. I'm going to finish with my favorite slide, because this slide is the first slide that I've ever generated through AI. So this, ladies and gentlemen, is a tree of knowledge in Quell's corporate colors. Isn't the future wonderful? And is really trying to reiterate the message that these feedback loops, as I've termed them, but basically this knowledge management, this has to be company-wide. So the outside of the tree, agnostic of function, I don't mind if you're in PD or immunomonitoring or clinical, or GXP or GMP or whatever, these are the things that you should be doing well. So you're training your people, making sure they know what they're doing really good investigations, getting really curious with some of the stuff that you don't know, continuous improvement, managing your knowledge, making sure that you're comparing apples with apples when you're looking at data from different functions in your company. This stuff is company-wide, and you can build this in right from the start, right? And Donna illustrated that absolutely beautifully. And then inside the tree, you've got the little kind of branches which focus on your product, your supply chain, your platform, your materials, your equipment, all that kind of stuff. And then you've got all of the branches that go around your clinical side, but they're all connected, right? They're all connected inside this lovely kind of tree of knowledge. And I'm going to finish by just walking you through an example of the difference between compliance and knowledge. And this comes to, let's say we're going to do a material receipt. So you have a material that you use in your process. It can be for cell selection, it can be for expansion, it can be for anything you wanted to do. And you're going to receipt it in a nice GMP style because you're manufacturing for a patient. So it comes into the warehouse and it has a C of A and it's on your approved supplier list and tick, 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 tick. You're not going to test it. No, it's early phase. Why would you do that? Goes into your warehouse, goes into your manufacturing product. Lovely, compliant, brilliant. Tells you absolutely nothing about that material. It may be that actually that material only works in a very narrow operating range for your process. And that's fine. This is the kind of knowledge you've got to build in. But when you are doing a root cause investigation, when you have a batch fail and you're doing your little 5 whys and your fishbone, because that's what you put in, because that's the right thing to do, it's compliant, for heaven's sake, please don't tick off materials as totally okay, because you don't know they're okay. You're compliant, but there's no knowledge. So I think the last kind of plea to all of you to say, from the start, focus on knowledge. These are the people at Quell. They are as brainy as they are beautiful. And this is obviously a massive team effort. So massive thanks to the guys at Quell, manufacturing, QA, PD, AD, MSAT, clinical immunomonitoring, the whole kit and caboodle. Thank you.
Thank you, Kasia. The last presenter today is Scott Jones, Senior Vice President and Chief Scientific Officer at BioBridge Global, who's going to talk about potency assay development. If you're not early, you're late.
All right, good afternoon. Like was mentioned just a second ago, I'm going to talk to you about potency assay development and specifically really how to do it early and, kind of with those guidelines. That way you're able to really make sure that you kind of get it right from the start. So with that, ooh, there's a giant arrow. Okay. And so with that, let me go over a brief outline. I'm going to go over potency assurance. Then I'm going to talk about assay life cycle. Like Donna, I'm a planner. And so honestly, if you plan, it's going to go a lot better. So we're going to talk some about that. And then specifically about design and qualification of that life cycle. Phase appropriate validation, which was already mentioned, give you an idea of what a timeline looks related to potency assays. And then talk to you some about the different assays, potency assays that we've developed in kind of our approach at BioBridge Global, and talk about a novel platform called Primiva, patent pending, so I can't go into a lot of detail, and then kind of give you basically a brief summary. So with that, let's talk potency. I'm sure most people here know what potency is or have an idea. It's not a novel word these days. But the bottom line is we did have the original guidance in 2011. The definition is shown here. Honestly, I don't like it. It's legalese. I still don't really kind of understand what they're trying to tell me. So don't look at it. The 2023 guidance is much better in my opinion. And that's really where they talk about a specific ability or capability to achieve the intended therapeutic effect. And that's really, in my opinion, that's what it's all about, right? It's what's the biological activity that your therapeutic's trying to do. Much easier definition. The other thing that's kind of cool in the 2023 guidance is they go over assurance strategy. So in other words, not just talking about potency testing, whether it's release, whether it's in process, but kind of what people have been saying here already, making sure that you have manufacturing process design and process controls and things all the way along the way so that you're basically able to show that potency. The other thing is, you know, if you don't know much about potency assays, is obviously, like I just said in the definition, it's going to be specific to the therapeutic. So the bottom line is there's no one size fits all, right? This isn't like a lot of your safety assays or types, things like that. The other thing is there's going to be some level of assay development, right? Because it's specific to the therapeutic, thus you're going to have to probably develop something or multiple assays for it. So in other words, it's not plug and play. So the question then is, how do you develop these things? How do you come up with them? And how do you what kind of process can you use to make sure that you get it right from the start? And so one of the things we implemented, and I highly recommend, is really to generate an assay life cycle. And this is based on USP 1220. Obvious is what we use, but you can put your own flavor onto it. And I'm not going to go into all these details because that would take like hours. But the bottom line is it's broken down into 3 parts. You have your design, your qualification, and your maintenance. And really the take-home message here is there's a lot of stuff listed here. This is a complicated and going to be a time-consuming process, right? But if you do this right and you do these checks, like risk assessment and the right qualification and validation, I'm telling you, in the long run, it is going to save you time and make sure that you don't have to do rework. So the bottom line is, and the mantra for my talk is, start early because of this. The next thing is looking at assay design. Once again, not going into the details. This is basically taking it out of the diagram that I just showed. But really, it's kind of what was already mentioned. This is knowledge gathering. Knowledge is power. And so the more you know about your therapeutic, the biology, the mechanism of action, the instrumentation, do you really want to automate? What should you automate? What shouldn't you automate? Those type of things is really what you got to think about early in the process. The other thing that I highly recommend is a risk assessment. Kind of what was already mentioned, whether you do an FMEA or a fishbone, whatever your style is, I'm telling you, it will save your butt. So a good example I have is we were working on a flow cytometry assay for a customer. And luckily, we have a very active quality department. They're like, Scott, you got to do a risk assessment. And I'm like, okay. So we did one. And sure enough, this one factor or this one thing came up that we had a high probability or a possibility of like cell clumping. So we figured out what to do. And sure enough, it was an issue. And so we were able to address it before it really became a true issue. And so because of that, it didn't affect our qualification or eventually the validation of that assay. So I'm telling you, it will save you. So, and then the other thing is, especially with potency, you're probably going to end up with multiple potency assays. So the other thing I would recommend and what we've been able to do is I would start with several potency assays. And then as you go through and start going into your phases and after you qualify, then you can decide, what do I truly qualify? What do I truly validate? And so more than likely, you're going to start with quite a few and end up with just a few, but these are going to be the ones that you have determined are actually the true or best indicators of potency for that product. And then once again, the mantra, start early because it's going to take some time. And then the next thing is qualification. If you look at the bullet points here, these may read almost exactly like what Donna showed earlier. These are really kind of the same verbiage. And the bottom line is because of limited knowledge of the product manufacturing, really do phase-appropriate validation strategy, really that just in time. And the other thing is early in development, it's okay to do the fit for purpose. And then later, once you have more knowledge and understanding, then go ahead and do your true qualification or validation. And one more time, start early, because this is obviously going to be something that takes time. So to give you an idea, it's not it's not AI generated, but this is my take with some help from friends on sort of an idea of showing you kind of phase appropriate validation timeline. So what do you see? What you see is over in preclinical and even before that, you've got lots of different assays you're looking at, right? Start with many. And then what you can see is you start for fit for purpose, then you go ahead and qualify those assays. Some of those you may then start using in phase 1 and 2, but then you may discover as you generate more data and you're looking at your data that actually it isn't a true indication or a good indication of potency. Whereas then eventually you do have some that are. Those are going to be the ones that you then validate and are what you're going to use later on for phase 3 and then commercialization. Okay? Make sense? So, what do people do like myself and my group at BioBridge Global if we're assay developers and we're creating potency assays, but we're not therapeutic developers? So there's kind of a bit of a conundrum, right? Because I just stated earlier that these assays are going to be specific to the therapeutic. So how do you do that? So once again, if you lean on the concept that potency is really biological activity, so that's what we did. And as you can see here is we went through the process outline, but really we looked at biological function. So we created custom potency assays for the different functions and what I'm going to call scaffold assay, right? So we have some for cell killing, for immunomodulation, chemotaxis, angiogenesis, what's mentioned. And so the concept is that way we've done a lot of the early design work, and then we can work with the actual therapeutic developer and come in and say, hey, based on this, these are the ones we recommend. We can now customize it, use whatever cells, change incubation time, whatever you need to do. And then that way we can qualify it easier or possibly even validate it quicker. But that way it's shortening that time and kind of decreasing that true, use on early. In other words, I'm trying to save some of the time up front. And so far that's been very effective. The other thing is we kind of have a general philosophy, and that is we try and make our assays to mimic physiology as best as possible. We like to miniaturize. Why? Because miniaturize makes it easier to automate. It also allows you to use very small volumes, so you're only using a few microliters. And that way we can quantitate and also make it highly customizable. So with that, one of the other things that we've done is we created a novel potency platform that can also be used for other things to understand mechanism of action and other things. And it's basically called Triumvira. It stands for Physiologically Relevant Miniaturized In Vitro Assay Platform. And it is patent pending, but let me tell you a little bit about it and how it can kind of help in these situations. And so what it is basically it consists of 3 parts. You have a culture vessel. So in the culture vessel, you can grow different cells. As you can see on the slides here, I don't know how well you can see in slide A, but basically we're growing lung carcinomal epithelial cells, right? And they've been tagged so they're glowing green. Then what we do is we put this culture vessel in basically a circulatory system. So it is flowing through this vessel just like basically mimicking circulatory blood flow. So it has the same shear forces in that you would see in a human body or human physiological condition. And then what you can do is then you can add in whatever your cell of interest. So in this case, if we're doing cell killing, we're throwing in T cells that have been labeled red. And then the third part is for current detection, what we utilize is a high-speed camera. So what you can do then is as these T cells are flowing through in physiologically relevant conditions and pressure, they basically will then interact. As you can see, if you look across A to E, and as time goes, you actually see the green disappearing, more red, it's attaching. You can actually, with the camera, you can see them grab and roll and all kinds of things. So you can either look at it live or, like here, you can just look at it and get an endpoint type of assay. The other thing that's kind of cool about it is, well, what's going on with the T cells? Well, you can actually take samples out, monitor those T cells, do whatever characterization you want, look at whatever surface markers you want, and you can also look at media and see what's being secreted. And so the same concept is you could do it with CAR T cells or CAR NK as shown here, and it's really kind of the same thing within the culture vessel. So what does this actually provide? The other nice thing is it's an open platform kind of. So in other words, it's cell type agnostic. So I don't care if it's an allo or a auto-engineered cell, it'll work. And then you can really use it for identity, specificity, and even potency. The other cool thing is we're starting to do some studies where you can actually, like was mentioned, the more info you have, so you can do studies to determine mechanism of action. You could also do therapeutic repurposing. And the other cool thing is, think about it. You have a culture vessel with one type of cell. So what if you have multiple culture vessels that you put on a small chip and you do different organs? So you can do lung, you can do kidney, you can do liver. You can basically, in fact, we're in process of daisy chaining it together. So it basically is almost an entire human physiological system on a chip. So that way, if you have a therapeutic and it's supposed to mainly just target at certain cells in the liver, that way you can also run it through a system like this before animal models and actually show, hey, is it interacting with these other cell types in a close physiologically relevant system? So it has that ability to do it too. So bottom line is, you mimic circulatory blood flow and vasculature. We're showing, and I don't have data today, but you can actually look at adhesion molecules and you actually see a different expression of on and off of some adhesion molecules because it is in basically having the shear forces in the circulatory system. The other thing is miniaturized, so volume-wise, you're talking 100, 150 microliters are what we predominantly use. And then the other thing is it's fully customizable for the developer. The other thing that's kind of cool, so you're like, okay, that's kind of nice, but you're having the label cells. And really, we're trying to get to a label-free system. So we actually have done a couple of runs now, and we basically have a system where we can actually use impedance-based monitoring within the system as it's working. So you can basically then do it without having to label either your target cell or your therapeutic cell with that. So bottom line is what, let me give you a brief summary. So I know I'm the last speaker. I know it's after lunch. So if you've been dozing off, wake up. Here's kind of the take-home messages, right? I'm trying to make it easy for you. Basically, advanced therapy products will need potency assay development. Don't fight it. Work on it. Work on it early. Start potency assay development ASAP. Like I said, if you're not already working on it, you're probably in trouble. Use an assay life cycle. Go through that process. Do your risk analysis. I can't stress that enough. And also develop multiple candidate potency assays. And as you go along, validate or qualify those assays as you go, ending up with a couple that are best. Do phase-appropriate validation and whether you do it yourself, and these things all apply, whether you're doing it yourself, you're having us or some other assay developer do it for you, but there are assay developers there to help you. So you can focus on your therapeutic if you need help for these type of things. So with that, just wanted to say thank you for your time. You can come visit us at BioBridge Global Advanced Therapies at booth 376. Be glad to talk to you about it more. And I'm just the mouthpiece. The cool, smart people are the ones listed below here. These are the ones that really developed the system and did all the work. So with that, I'll say thank you. And I'm right on time.
Donna, David, Kasia, if you want to join us.
I have a couple questions for the panel.
They have no backstory.
The first one, and you can all choose whoever wants to start. As technologies in manufacturing and analytics evolve rapidly, how do you recommend building flexibility into early development to allow for integration of new tools or methodologies later on? Of course, not everyone has the money right from the start to go full big. How would you help the audience and anyone here? Who wants to start? Donna?
I go back to understanding what your core process is, understanding what your proposed mechanism of action is, and what you expect your product to be. What you expect it to be may not be what it is. And so that's where understanding all the assays, the knowledge base, and the potency development plays a role. But how do you, as a company, you started out, you do this, and here comes better ways potentially of doing things. And that's where I go back to building a base of, I live and breathe by those internal process controls. I have source material from the day we started, from repeat leukapheresis on donors that I manufactured the product with our initial process. You went from, version 2.0 to 2.7. It's the same thing. I know what it did at this stage. I know the characterization. I know my potency matrix and what data I got from that product at that level. And as you go through, it's one thing if you're constantly having to get new source material to work with, that comparability and being able to transition, appropriately assess new technology, new instrumentation becomes much more fluid and easy if you've taken the time to build that base core control material that you can take all the way through the process.
Nice. Anyone to follow up on that?
Just ditto, really. Focus on the biology and lose the ego. It can be awful when your pet project gets canned.
Yeah, I think you try and choose, kind of like what was mentioned earlier about making the right choices from the beginning. So I think part of it is trying to be as flexible in the beginning as you can. Make sure that what platforms you do choose are not crazy narrow so that you do have that possibility. Because like you said, once you get into it and start really looking at things, especially from an assay standpoint and qualifying, a lot of times you don't end up with what you think you're going to end up with. So you need to make sure that you do that. But I think part of that too, and that's the same thing even when new technologies come around, I think you have to really do a proper assessment and don't just jump on the bandwagon, but hey, is this truly going to be a benefit? What is the benefit? And if so, then how do I implement it like was mentioned here by Donna? How do you then implement that in your different, phase 2.0, 2.1, whatever?
Yeah, before you open up the checkbook to write a big check for an expensive piece of equipment, make sure that it fits what you're doing. Don't feel pressured into doing that just because you feel like you need something that's automated or you're getting outside pressure on. It does have to fit the biology.
Thanks. Last question. For companies just starting in cell and gene therapy, what's one piece of advice would you give to ensure that their early decisions set them for long-term success? One each. Kasia?
Make sure you have got the advice of somebody who knows what the end is going to look like.
Remain flexible.
Work on potency assays as early as possible. It's the same thing I just said.
Know what's required from a regulatory perspective.
Yeah.
Yeah, that's good. That feeds everything.
Yeah, she wins.
All right. Let me thank you for your time and your participation, and please give them a round of applause for me. Thanks, everyone.